briefly. So, okay, so this one's about immunity, infection, and inflammation. And just again, general overviews. You'll need whole courses to really get into the what? I don't know. Oh, itis. Yes. We'll get into that. You're jumping the whatever line. All right, so yeah, we're going to go over its background, talk about inflammatory mediators, chronic inflammation, and then some of the adaptive immunity as well. Again, the reason why this is important is because a lot of pathology, pathophysiology, is mediated by your immune system freaking out and starting to destroy stuff that should not be destroyed. And we've already seen this. Maybe we haven't that much. I don't know. But like all your rashes, right? Those are mostly 99% of them are because it's your immune system freaking out because it was exposed to something. Whether it be like a sunburn or like a poison ivy or whatever, that's all immune regulated. So the immune system is so important to this class. All right, so we're going to be talking about the adaptive, innate versus adaptive, and then cell mediated versus immune versus humoral versus physical. So this is kind of a nice little chart that summarizes everything. You might have even seen this before, but immunity is generally broken down into innate versus adaptive. Innate being things that are fast acting, ready to go, but have less of an effect on whatever that pathogen is or stimulus. And there's the innate that's always on, and that's like cells that are in your tissues that are ready to go. And that if you get an infection, it's good to go. These are things like your tissue resident macrophages. Well, and then there's the immediate response things. I guess that's more like your tissue resident macrophages and the immediate response. Things that are always on are things like your barriers, like your skin is part of your immune system insofar as it prevents infection by providing a barrier there. It's always there unless you have like a cut, I guess, but like that's a different story. And then things like your complement system, which y'all might have heard about as well, but we'll go into briefly. This contrast to the adaptive system, which is more delayed responses, where you have things need to develop. You need to develop a robust T cell or B cell to generate antibodies that can target these things specifically. And what we'll talk about is that once you generate these cells, really what you're doing is you're really arming your innate system to really destroy whatever's there. But we'll talk about that more a little bit later. So the first line of defense is things like your physical barriers. These are like your skin, your linings of your gastrointestinal tract, your whole alimentary canal has all these mucus secretions. And we talked about that with cystic fibrosis. You want to have like a healthy layer of them, but not something that interferes with the physiology of it. But that mucus barrier is something that pathogens would have to get past for things to really invade your body, right? And so that's part of your immune system as well. Even neurogenic responses like coughing and sneezing, where what you're doing is you're physically removing anything that got in, and things like your stomach acid, really acidic environments, pathogens that are in your food, which are absolutely there. A lot of people there's microbes everywhere, right? So if microbes are in the air or just randomly land on your food, you absolutely eat stuff. The thing is that it's so acidic in your environment that most of the time, as long as it's not a pathogen that overcomes it, like an E. coli outbreak or something like that, most of the time that stomach acid is just going to incinerate whatever pathogens are there, and then it's going to be fine. Then there's also biochemical barriers, so things that are exposed to the external environment, like your mouth or your your eyes. We have things like saliva and molecules in your tears that contain antimicrobial peptides and different things that break pathogens down to where whenever they land on your eye or land in your mouth, they can even get eliminated before they even reach something significant. These are secreted. Earwax is the same way. There's also stuff in your sweat that are antimicrobial to delete stuff. Or to get rid of stuff. There's also your normal flora in your intestines and on your skin and stuff like that. That's also part of your immune system insofar as these bacteria compete for access to nutrients with pathogenic stuff. But these bacteria are either helpful or don't really do anything and are commensal to your gastrointestinal tract, and they just live there and either produce vitamins or don't do anything. But anything that's pathogenic, like an E. coli, like a pathogenic E. coli, it doesn't have access to the nutrition that it needs to really take over and cause severe disease in your body because it's competing with your normal flora. Does that make sense? So it's considered part of your immune system insofar as it can help prevent infection, help lower the probability of infection. So when we look at molecular stuff, there's also the complement system. Have y'all heard of this? This is like something that's not as extensively taught. So the complement system is a collection of proteins. They're almost like innate antibodies, but not really. What the complement system is, it's a collection of molecules that are secreted by your liver that can help bind to and get rid of pathogens in your bloodstream or in your tissues. And then there's also cellular defensins that can contribute. There's things like lysozyme, alpha, defensin, different molecules that can just help kill bacteria that are proteins that are made by your body, right? That are part of the innate immune system. So what happens is that if you're a pathogen, you got to get through those physical barriers first. Then secondly, get past these chemical barriers that are made to kill you. And then even if you do that, your body then has the ability to trigger an inflammatory response. And what this is it's the explosion of your immune system at a specific localized spot. Or there's also general inflammation, I suppose, but it's an explosion of your immune system at wherever this pathogen is. That it's a robust multifactorial approach to getting rid of whatever that pathogen is. And what I mean by that is that the first thing that'll happen is like you get some cut, pathogens will go in. You have the restriction of your blood vessels, the recruitment of your white blood cells in your immune system to the area, different tissue repair systems get triggered. All of that's associated with inflammation. And how it manifests itself is with redness, heat, swelling, pain, and loss of function. So we've all experienced this anytime you've had a hangnail or whatever, right? Where it gets red because you have increased blood flow to that area. That's increased blood pressure so that your immune cells can be recruited to it to fight whatever the infection is. There's heat. That's just increased amount of friction because you have increased blood flow. So that's kind of tied to redness. There's swelling. Again, you're increasing the blood flow to an area. Well, that blood has volume, and so things are going to swell up and expand. There's pain. That's like a molecular trigger that acts on your nervous system to help regulate that you don't use that area. Because if you cut your finger, for example, you don't want to be consistently using your finger and then like normal and then be picking up things and exposing yourself to more pathogens potentially. You want to be like, ow, this is hurting, and then like get it away from everything so your body can heal it. That's actually like a selective evolutionary response to stuff. Pain is, so it's a good thing. And then tied with that pain is a loss of function. Your body's going to turn off whatever it is. I had a dog bite me like a couple months ago, and like it was weird that like I just couldn't move. It bit me right here, right on the middle finger. I just couldn't move the middle finger, even though I tried to. And it's not because it like cut a nerve. It was just like paralyzed almost. It felt weird. Whatever though. That'll be a good example dog bite for this lecture, I suppose. So yeah, there's this vascular response, blood vessel dilation, increased vascular permeability, leakage, and white blood cells, extravasation. That's just a fancy word for the blood cells coming out of the blood and going into the tissues, extravasation. Okay, so inflammation is a complex, non-specific response to tissue injury. It can be acute or chronic. Chronic meaning it's long-term, has lasting effects, while acute being short-term and has specific targeted effects. Inflammation is a normal part of the body's defense, important component of the innate immune response, and is set stages for the adaptive response. Right, so inflammation. This inflammation is a part of the adaptive immune response insofar as it's generally trying to get cells in an area to help protect against pathogens. And this response is not tailored and specific to what those pathogens are, which contrasts to the adaptive arm. Does that make sense? Okay. One of the first responses, yeah. And so what this inflammation does is it aims to removing injury. It wants to remove the agent. And so what happens is that it's going to be relatively successful. The only thing is that if the pathogen is very good at being a pathogen, it's going to overcome these innate inflammatory responses. And so that's why we've developed the adaptive arm to help protect against things that are better than our adaptive immune system. It's this like fight between the pathogen. It's this evolutionary fight between the pathogen and the immune system about who's going to survive better. So yeah, this is what you were saying. So these inflammatory conditions are named by adding the suffix itis to the affected organ or system. Yeah, and so it's an easy way to like just generally name stuff. I used to talk with these high school students, and one of them walked up to me and was like, "You know what bronchitis is?" And I was like, "Yeah, it's like inflammation of your bronchioles." And they were like, "Man, how'd you know that?" I was like, "It's like you don't get like I get you're a freshman in college, but it's like this is like basic stuff." So yeah, it's really important. Yeah, so itis, that's an important thing to. I think that's a test question even, that it's like, what is, I think it might be bronchitis, right? What is bronchitis? And it's like inflammation of whatever. So yeah, we're going to be talking about some of the immune cells. Again, this handout's online that you can go over specifically if you need a refresher on anything. And I recommend just giving it at least one, because there might be stuff on there that you didn't even know you didn't know. So just give it a look over briefly. And then if that's all you need, then you can. But I think there is a question on there, and I think it involves like it's a question like it's something similar to like, is a neutrophil a granulocyte or not, or something like that, where it's like you need to know your classifications of your cells for the test. Okay. Okay, so, but briefly, what we'll talk about briefly about phagocytosis. Phagocytosis is just where a immune cell is eating something and it's trying to target a pathogen for destruction. We consider it eating because it comes around and it forms this little vesicle around it and infuses the vesicle with the lysosome, which is an organelle that's very destructive. And so when it infuses it with that lysosome, it destroys all little pieces and parts of the pathogen, and it can move on, and then that's a way that your body can remove stuff. So we have a couple different phagocytic cells. The most commonly seen ones are dendritic cells and macrophages. And what's great about these is they digest this pathogen, and then they can present it to the adaptive arm, and then that's how we get adaptive responses to B cells and T cells. And in this lysosome is this really effective set of molecules called reactive oxygen species that are very oxidatively damaging, and that's how it destroys this stuff. It's actually funny. People used to take. We always see antioxidants as like a health benefit, and there's this whole like little history with a guy named Linus Pauling, who he discovered that people who ate blueberries didn't get as sick as much, and he like tried to target it down. He thought there was something about the blueness of it, and he targeted to these set of aromatic chemicals that have antioxidant properties, and he thought, oh, antioxidants are good for people. And then we got this whole narrative about antioxidants being super good for you and how we want to consume antioxidants. But turns out people concentrated specific antioxidants, took those as like a medicine, and found that people got not only more infections but also more cancers. And what they found was that what they're doing is that they're disrupting this process fundamentally, that if you take so many antioxidants, you can interfere with your lysosome. And so what pathogens can do is they can just progress through this, not be damaged, and then like can still replicate and stuff like that. And they found out that it was really other vitamins and minerals of the blueberries that were making people healthy, and not the antioxidant properties. But I'm not saying antioxidants are bad for you. Don't get me wrong, but it's definitely like a level of like you don't want to take excessive amounts of antioxidants.
It seems like too much of anything.
That'll be a general trend throughout the class. So I'm glad you're recognizing that. But yeah, so that's an interesting little story. Regardless, though, there's also neutrophils, which are innate cell that phagocytize stuff, and then they have a very, very short life, only last a couple days in the body. So they pretty much phagocytize stuff and then die, but they can stimulate inflammation and propagate it a little bit. Then there's also this cell-mediated recognition, and there's certain cells like natural killer cells and T cells that do this, and they recognize cells that have a problem. Because what's great about this phagocytosis is that this is really good, but not very good if the pathogen is already inside of the cell, right? Because this is only good for if it's something smaller than the white blood cell, and it can be phagocytized. But if you're already within a cell, right, like viruses are, or if you're a cancer cell, you can't really phagocytize whole cancer cells very well, right? So the immune system recognized this and then developed this whole separate cell-mediated way of killing stuff, to where if you're a virus and you're actually living inside of a host cell, the immune system can still recognize you and kill you with these cell-mediated response, and it's regulated by these different biomarkers that are found on cells, there's different activating receptors that activate the immune system and inhibitory receptors that stop the immune system. And if you're a healthy cell, you have an inhibitory cell to where you tell the immune system, don't kill me. But then if you're an activating ligand, you can tell the immune system to kill me. What's interesting is pathogens recognize this and then regulate it extensively. And there's this whole battle between molecules about what's going on with your immune system. But we won't go into all of that this semester. Okay, so extravasation. So I mentioned this a little briefly earlier. It's this whole process of like sticking, rolling. There's all these proteins involved. There's all these carbohydrates involved. You don't have to worry about that. At the end of the day, what I want y'all to know is that leukocytes, which are white blood cells, they're in the bloodstream, and then they can be recruited out into the tissues where the pathogens might be, and then they can search and destroy these pathogens that are in the tissues by getting out of the blood and going into the tissues. There's also cytokines, which are like chemical messengers. This is how the immune system communicates from one cell to the other. And they can have pro-inflammatory, which means you are supporting inflammation so that you can affect stuff, and anti-inflammatory, where you're trying to turn it off because you've already eliminated the pathogen. This is a regular thing that you get a cut, you have pro-inflammatory mediators be released, you have inflammation, the inflammation deals with whatever the pathogens are, and then you turn on anti-inflammatory mediators to turn off the inflammation, and then you resolve and you go back to a normal physiologically stabilized homeostatic state, right? And so there's different pro-inflammatory and anti-inflammatory effects depending on stuff, and there's all kinds of molecules like tumor necrosis factor, interleukins that you might have heard of in the past, but I won't make y'all learn specific ones, at least for now. I think you have to learn like one or two of them for the test, but we'll get to that later. So there's also acute inflammation, which is just inflammation at a very localized spot, and then this contrasts to chronic inflammation, which is very problematic in things that you see in like heart disease or cardiovascular diseases rather, that you have inflammation across your entire bloodstream pretty much, and well, that's going to really, really increase your blood pressure and cause all kinds of problems, and that is what atherosclerosis will develop from that, and we'll talk about all that later. But that's another condition about why you need to know the immune system so well when you're talking about pathophysiology. So there's different stages of acute inflammation. You have two different seemingly contradictory responses whenever you have acute inflammation. And I don't know, can y'all see that here? So what'll happen is that if you scratch yourself on like your skin, what you'll see is that there's like this red streak down the middle, but it's surrounded by flanking 2 white streaks. You guys kind of see that? So what's happening there is that the red streak is increasing the blood supply and recruiting in blood cells, because that's directly where the damage occurred, where the pathogens can get in. So your immune cells are trying to get in that really red spot, but your body's smart in that it shuts off all of the blood flow around that area, to where if a pathogen did get in, it can't spread to the bloodstream very quickly. Does that make sense? So it's allowing your immune cells to get in, but preventing the pathogens from spreading out. And it does this by recruiting in blood vessel, by increasing blood vessels, or increasing blood pressure here, but decreasing it here, right? By just shutting off blood flow. So there's whole vascular control of inflammation, as well as a cellular stage where you have migrations of the leukocytes into a specific area. And this is mainly neutrophils, which are the first responders of the immune system. Those are great because they can do phagocytosis and they have cell-mediated roles as well. You recruit in the cells that you need of the immune system to really effectively have a good, at least good, acute inflammatory response. Good. This ends with leukocyte activation. So what all of this is trying to do is fight the pathogen first and foremost, but then secondarily take that pathogen that you're fighting and present it to your adaptive arm so you can start to develop T cells and B cells for the adaptive response. So I think, there's immediate transient response, minor injury, rapidly usually reversible, short durations, immediate sustained response. This is more serious injury. So you can have different levels of vascular responses. Obviously, like if you have a small little paper cut, that's going to get resolved pretty quickly. But if you have a full-on dog bite, that's going to maybe take a couple days to really resolve all the tissue damage. Maybe, I guess, weeks to resolve the tissue damage, but then days to really resolve all the microbial exposures that might have happened. Delayed responses. That's funny. I have dog bites in here. That was before that happened, I think, at least. Delayed responses. So there are certain delayed responses that, like, you can be exposed to sun radiation. Apparently, this is not a thing for Black people. This blew my mind whenever I learned it. I was like, I have a friend, this is Black, and I was like, oh yeah, I got a really bad sunburn. He's like, can't relate. And I was like, what do you mean you can't relate? And I was like, Black people don't get sunburned. And I was like, that's awesome. She just straight up said we just get darker. Like, it's like all we do is tan.
Yeah.
Oh, it absolutely. But it's just like, I don't know. I just never heard of that before. It's like happened like a few months ago. Yeah. Anyway, like what'll happen is that this radiation will hit you. It'll mutate all these proteins in your DNA, but nothing's problematic yet because everything's concealed. It's not until that cell dies and then releases pro-inflammatory mediators, which is like 1 or 2 days later after you get the sun exposure to the sun, where you have a response. So that's why there's a delayed response of the inflammation, because you haven't activated the immune system. It's not the immune system that's delayed, it's the exposure of there's a delay between the exposure of the event and the exposure of the molecular patterns that trigger inflammation. That is really why you have a delay in that inflammatory response. But yeah, your sunburns are just generalized inflammatory responses that's why you're all red and stuff like that. It's all immune mediated. It has nothing to do with like the heat or the UV rays that cause that redness. It's just your immune system, which I thought was interesting. So you activate your immune system. You have leukocytes that get recruited, the first being neutrophils, and those, but also monocytes will recruit from the bloodstream and go into the tissues and will turn into macrophages. What's great about monocytes and macrophages is they're really good at phagocytosis, just like neutrophils are, and they can recognize stuff, engulf them, and kill whatever pathogens might be available. The problem is that, so you have with acute inflammation, you can have potentially complete resolution of whatever that thing is, and it happens a lot, right? It's like you don't need B cells and T cells to heal a paper cut, right? You only need B cells and T cells to help with like COVID, right? It's like you can have complete resolution um, of pathologies like a cut with your adaptive arm. However, there might be some damage insofar as there might be scarring and fibrosis. Like I got a scar from you can't see it from there, but I got a scar from this dog bite. It was actually funny. I went in like to this urgent care place, and one of the questions they asked me was, "Am I a hand model?" And I was like, "What? You have to ask?" Yeah, a hand model. Like, do I model my hands? And I was like, "Thank you. This is a compliment, I guess." But no, I don't. It was a weird question, but yeah, yeah, it was. Well, she was like 60. I don't know, maybe so. Yeah, you're right. Anyway, so you can have scars that happen, and then you can have progression to chronic long-term inflammation, which does happen occasionally and I think we even see some of these conditions later on in the semester, when it's like it started off as acute and then it never got resolved and it just continued for forever and turned into chronic inflammation. Happens a lot with chronic kidney disease, CKD. When the offending agent is not removed, yeah, that happens. Okay. All right, so we have the acute arm comes from a couple different areas. The first being plasma-derived mediators. So these are things that exist in your blood that help generally fight everything. So this is stuff like your complement system that I mentioned earlier. These are made by your liver that help fight stuff. And one of the important things, and I do want to note that these are bolded because I think, if I remember correctly, this was like a question is like you knowing which of the following is a plasma-derived mediator type of thing. And I think the answer was actually C-reactive protein, but I can't remember. I need to look at those test questions before I lecture, I guess. But yeah, make sure you know the plasma-derived mediators, C-reactive protein, coagulation, vasodilation, and vascular dilation permeability. And then know the cell-derived mediators. And the only ones that I want y'all to know for this is IL-1 and TNF-alpha. So just two of the cell-mediated ones. And both those are strongly pro-inflammatory, really good at fighting pathogens, whether it be bacterial or viral or a lot of stuff. A lot of really good antiviral properties of those, though. So for the plasma-derived mediators, here's some examples. So again, these are synthesized in the liver. They're synthesized as precursor forms because you don't want to always have these things active because then you'll have destruction all the time. You only want to have them active whenever they're exposed to whatever the target is. So they're synthesized as a precursor and then can be quickly activated if there's ever an exposure event to whatever the pathogen might be. And here's some examples: acute phase proteins, complement system, coagulation system, the kinin system. Same thing with enzymes. There's pro-enzymes that need to be activated that can have antimicrobial effects. Substrate of the activated enzyme becomes the next component in the series. Yeah, I don't really need to know that, but yeah, these are really good reactions to things that happen. So the synthesis of these proteins is stimulated by cytokines. So this makes sense, right? If you have something made in your liver that's antimicrobial, you get exposed to a cut, your immune system gets activated because you're responding to the cut. Your immune system is going to start releasing cytokines. Those cytokines are going to travel through the bloodstream to the liver, and you can make antimicrobial stuff. So that if the pathogen does get in past that, your blood vessels constrict and then don't allow things to get out into the bloodstream. Let's say a pathogen does, though. What happens is that you've activated a system that's all over your blood that can attack any pathogens that are circulating in your blood, and it's really great antimicrobial stuff. But all that's stimulated by the cytokines that are released at the spot of infection. IL-6 is another thing that's produced by macrophages in response to infection, and it's one of these things it's one of the cytokines that is released that acts upon the liver to produce these acute phase proteins like C-reactive protein. That's just IL-6 is an example of that type of cytokine. Yeah, you don't really need to know that picture, and just know IL-6 is an example that acts on the liver here. So complement system, yeah, like when I was taking my immunology class, we had to memorize all the 3 complement systems and know all the differences between it. Y'all don't need to know that. Just know that these complement systems, they are a collection of proteins that are released by the liver that induce inflammation and can destroy pathogens directly. And then it also can activate and with other parts of the immune response to help fight pathogens as well. But there are technically 3 different pathways that get triggered by different stuff and act different ways. You don't need to know that. There's also the coagulation, which we're going to talk about extensively whenever we get to the blood clot and wound healing lectures. But it forms this fibrous meshwork at the site of injury, and this is to help healing. Because once you've eliminated all the pathogens, right, you got to start healing that wound after the cut, and this is the way that you do this. And there's a really important protein called fibrin that's really instrumental in doing and in forming that blood clot. And these factors that kind of induce these clots and the fibrin formation, that's what we were talking about in the genetic disorders. Those hemophilia people that like can't form blood clots. This is what we're talking about. That they can't initiate this site, and that's why there's just excessive bleeding and bruising. So the kinin system is really important for your inflammatory response as well. There's a kinin called bradykinin, and it causes dilation of the blood vessels. It's part of your pain response. This is one of the things that act upon your neurons that make you feel pain. It also causes that smooth muscle contraction that restricts the movement of pathogens out of the blood into the body. And increases vascular permeability at that site to increase blood recruitment and the recruitment of these leukocytes into the damaged area. So there's all these other ones as well, cell-derived inflammatory mediators. I just want y'all to know some examples of them and just be able to recognize them. Y'all don't necessarily need to memorize all of what each one does. I think that's what I'm getting at here. But just know things like reactive oxygen species, nitric oxide, platelet activating factor. Those are all great examples of cell-derived inflammatory mediators. There's also the arachidonic acid pathway. The problem with this lecture is that there's just so many little pieces and parts of the immune system that you just got to. What I want you to know is generally what the terms are and generally what they do. But there's a whole bunch of specifics behind the scenes about how these interact that it would take you like 3 or 4 different types of classes just to learn all of it. So it's very complicated, obviously. But the arachidonic pathway is another example here where it does a lot of the same stuff, where it's going to promote inflammation that leads to inflammation. Y'all don't need to know the arachidonic pathway if y'all want to get rid of that from y'all's lectures, but that won't be a test question meaning. So yeah, we talked about a lot of these already. So IL-1, TNF-alpha, IL-6. We talked about C-reactive protein has popped up a couple times. Serum amyloid protein A, you don't need to know really. I should have unbolded that. And leukocytosis and leukopenia. Leukopenia is a term that is from a lecture that we don't have to that we didn't get to go over, but just know that leukocytosis, which is that's increasing the amount of white blood cells that your body is making, so you can have a better immune response, which makes sense, right? If you have an infection, you want more white blood cells to help fight that infection. Fever, we've all kind of seen before. This is mediated by those same cytokines, IL-1, TNF-alpha, IL-6, another one that you don't need to know. But what it does is it acts upon the hypothalamus, and what it does is it upregulates it just increases your body temperature. And a lot of people think, Oh, that's to help fight. That's to help fight infections, right? Which makes sense. But really, what it's doing, it's not really fighting infections. Fevers are. It's really shifting the optimal growth temperature out of the optimal growth range of most pathogens. Most pathogens have evolved to um, replicate at 37 degrees Celsius, which is 98 whatever Fahrenheit. Um, and when you shift to 100 degrees Fahrenheit or 38 degrees Celsius, you have this fever and this pathogens don't grow as much. Additionally, your immune system also works a little bit better at increased temperatures because of this, right? It's evolved to operate at a better fever, to where it's just you're increasing the better probability of not allowing the pathogen to grow and increasing your immune system's response to whatever that pathogen is. And that's what fevers do. Shift the optimal temperature of these pathogens to destabilize them. They don't necessarily kill pathogens. To really kill pathogens, you got to go to like a lot, like 100 degrees Celsius, which you would be dead to, right? Yeah. So you don't want to. Fevers don't do that, right? All right. So there are differences between acute inflammation and chronic inflammation. So with acute inflammation, this is rapid onset. You increase exposure, increase vascular permeability, and you have the influx of inflammatory cells. It's relatively short, though. That's the big thing. Chronic inflammation is long. It's a longer duration. This lasts for days to years, and you just have constant exposure to pro-inflammatory mediators, and it causes all kinds of damage to the cells. There's this concept called inflammaging, where if you have generally exposed inflammation, you age faster. And the two best examples of this are people who are really stressed out at work. Like if you've seen the President of the United States aging pictures, have y'all seen that? Like, have you seen like Obama, like before his 8 years and after? He looks like he's like 30 years older, and it's only been 8 years. That's because you're very, very stressed as the president of the United States. And yeah, exactly, right. That's because when you're stressed, you produce a whole bunch of acute inflammatory mediators, and you have chronic inflammation. You age faster. It's also with smokers. People who smoke a lot, they look older than like what they are, and that's because you have a general inflammatory response to cigarette smoke, and so you age faster, right? Same concept there. Yeah, this is dumb. For chronic inflammation, it's self-perpetuating and may develop because of recurrent or progressive acute inflammatory processes from low-grade stuff. So it's just the fact that some chronic inflammation comes from acute inflammation that's never resolved, but some it starts off as chronic and then continues on as chronic. There's also these things called granulomatous lesions. These are granulatomas. I don't know if you've ever heard of these. These are like this is like you're exposed to something and your immune system develops a response to it, kind of walls it off. It's almost like a benign tumor. This kind of happens. I think those are called granulatomas, actually. They're walled off, but they're constantly causing inflammation because they're there, but they're not like bad in that they're walled off and like you lower the damage by doing that. And you have this kind of response to asbestos and different bacterial infections. With tuberculosis, it's super bad. We'll talk about that in the respiratory lectures, but it causes like problems being a granulotoma. It's caused by low-grade persistent infections or irritants that are unstable. Syphilis is another one that's on here. This is like a problem with like prostitutes insofar as they're constantly exposed to these STDs, and so they always have low-grade consistent inflammation because they're constantly exposed. But then they're fighting it, so it's like you're just constantly inflamed, and that's why you have this like pop culture depiction of like prostitutes as being like these old women and stuff like that, when like there's probably some inflammation going on there as well. So this can be all the pathogens, viruses, bacteria, fungi, parasites. There's also autoimmune diseases where you have an overactive immune system and it is constantly being triggered when it's just you don't really have any pathogens. And that's everything up until chronic inflammation, and we'll just end really quickly. We're going to go a couple minutes over, but I'll make it quick. Just about the adaptive immunity. So the adaptive immunity pops up whenever you have, it's after the innate response, but it pops up whenever you have a pathogen that can survive past the innate response. And this is your T cells and your B cells. Your B cells are what makes antibodies, and your T cells are a cell-mediated adaptive response to things. And so what'll happen is that your T cells are these cell-mediated response that deals with intracellular pathogens, so things that are inside the cell that are hiding from the B cells, right? And so you have different types of T cells, whether you're a helper T cells, whether you're a helper T cell, a CD4 positive, or a CD8 positive cytotoxic T cell. And what these helper T cells activate other T cells and activate the B cells, while your cytotoxic T cells, they actually do the actual work of killing infections. And B cells are the other type of cell type, which turn into plasma cells, which professionally secrete antibodies, these proteins that target pathogens, that remove stuff. So this is important because if you have an exposure to a pathogen, your adaptive arm will trigger, you'll have an inflammation, but then you'll eventually generate T cells and B cells that then will fight against whatever that pathogen is, to where if you have a second exposure like a month later, you'll have a very, very strong response. And that's because of the presence of these things called memory cells, because you have memory T and memory B cells that live for a long time and can help exposure. And this is why vaccines work, because what you're doing is you're artificially exposing your immune system to the antigens, and then you're developing a T and a B cell response, and then whenever you get exposed to that antigen, as a natural infection, there's a robust and strong response to the immune system, and vaccines are very effective in this manner. So it like primes the immune system for that exposure. And the timeline kind of corresponds to that. So, right, you need about 2 weeks for the B and T cells to be developed, 1 to 2 weeks depending on things. But then after they've developed, they then develop into memory cells, and then you have them for an extended period of time that range from about a year, where you have things like the flu responses that you need flu vaccines every year, right, to things like, I don't know, some other vaccine that you only need to take every, I think tetanus is this way, where you only need to do it every like 10 to 20 years or whatever the timeline is. It's because of different effectiveness of the T's and B cells. So yeah, B cells, they secrete antibodies. This is important. I want y'all to know what the B cells do, so you can kind of go through this chart, and what the antibodies do after they are made, and it's just in this chart. And these antibodies are secreted by B cells. There's 5 different types of antibodies. I just want y'all to be able to recognize the different classes. So IgA, D, E, G, and M. This is important for later on in the semester. We'll talk about what type of antibody responses are dealing with what type of things. So I want y'all to know that there are different types. Y'all don't need to know the shapes or anything. Just be able to recognize what the immunoglobulin, that there is an A, that there is a D, that there is an E, and that. So cytotoxic T cells target different cells, whether they're infected cells like with a virus or a tumor cell, and this is what actually fights against these things. We talked about those tumor cells a little bit later. Eventually what's going to happen, though, it doesn't matter if there's a B cell or a T cell response fully. What your adaptive arm is doing is it's trying to provide resources for the innate arm to destroy stuff. Your innate arm is the only way your body destroys stuff. The B and T cells just help do that, right? So if you're a T cell, it's not the T cell that actually kills stuff. What it does is it releases innate chemicals that induce killing of whatever that target cell is. Same with antibodies. Antibodies don't actually kill stuff. What they do is they provide attachment places so that different immune cells can come in, or a complement can attach and help destroy stuff. So that's an important concept to be aware of. And I think we'll just end there. host colonization infection, yeah, this is like, there's different microorganisms. There's extracellular and intracellular stuff. I just won't ask any questions on that. Different stages of infection. Yeah, this is all whatever. So yeah, we'll end there, and yeah, that's it. So any questions on that? It's just going to be a lot of information over the course of the semester. So